So today, I'm not going to speak about mass spec at all. I'm going to speak about one of the bioinformatic pipelines that we have in the facility. We have many, but Proteome Discoverer is one of the bioinformatic pipelines that actually allows us to analyze all types of data. It can do gel band analysis. This is straightforward protein identification. It can do quantification on data-dependent analysis, such as TMT projects. It can also work in data-independent analysis, where I've explained last week. It's a slightly different way of acquiring data with different window sizes instead of selecting for specific peptides for fragmentation. So the reason why I am kind of offloading you to Proteome Discoverer is because it's easier for you. I know that there's a transition phase in which everything looks very different and it looks very difficult, but it would be slightly, because we don't support you with bioinformatic analysis. Let's say we don't have a bioinformatician in-house yet. That's about to change. It would be very difficult for you if I was going to send you data in the very different formats, because each software is ideal for a specific type of application. For instance, if we are looking into DIA, the perfect softwares or the best softwares are Diane or Spectronaut. And then if we are looking into TMT analysis, Proteome Discoverer is probably one of the best ones, as well as MaxQuant. So if I would be sending MaxQuant and then Diane and not offering support for you to look at your data, it probably gets really complicated for you. So the reason why I'm kind of doing this in PD is because we don't have a bioinformatician in-house. Again, this is about to change. Right, so what I want to do today, and I'm just going to repeat this. This is a live demo of Proteome Discoverer, and I've chosen 3 different types of project: band analysis, an IP analysis, and a TMT set. Feel free to stop at any time. This gets more interesting if you ask questions and you're like, oh, can I search for this or can I look at this? Because then I can do this live for you. In the future, we'll probably have workshops, so you can come with your PC and you can do your analysis. But for now, I'm just going to present how PD works. In terms of what it looks at, so there's 2 main areas: identification and quantification. Identification is just the software will attribute a score to your spectra, depending how well it matches your peptide sequence on that specific spectra. And this is true either for DIA, which is data-independent analysis, and it's also true for DDA. They work very differently in terms of the algorithm that they use, because the data is also different, but in general, this is exactly what happens. So that's how peptides get identified. And the first level of identification is a peptide spectrum match, which is exactly that. It's when the software can see your peptide sequence attributed to that specific spectrum, and it attributes a score depending how many fragments have been identified, the intensity of these fragments, so on and so forth. And then the next level, which is actually depending on the type of project that you're using, if you're doing an IP quantification or if you're looking at the total proteome changes, the next step is quantification. And quantification it's also done slightly different depending on the method that we use. For instance, if we are doing TMT, in which we label each individual sample with a different TMT label, multiplex everything, and then fragments, quantification comes from the intensities of the reporter ions in the TMT. And basically the intensity of each of the reporter ions is going to let you know what's the intensity of that specific peptide in that specific sample, and then you can compare across the different samples, right? If you're doing label-free, what we usually do is we calculate the area under the curve for specific PSM or peptide spectrum match, or the peptide, and then we average or sum the abundances of the 3 most abundant peptides or all the peptides within a protein to calculate the protein abundance. In DIA, and this is in DDA, so in DIA, you actually have more information coming through. So you have the MS1 area under the curve, which is the peptide mass, but you also have different fragments. In some cases, you can have 20 fragments, 10 fragments. So you also do an area under the curve of the fragments of each specific that you can see in the spectrum, and you use the abundance of these to calculate the peptide. So it is the MS1 level, the intensity from the MS1, but also from the MS2. So there's more data coming through. So the quantification, let's say it's more secure. It's coming not only from the MS1, but more data coming through for the quantification. So Proteome Discoverer. There's many things that happen behind what we give you. So we usually do the analysis for you. And the reason why is because obviously this is a software that's commercial. So we hold a license, so we can do the analysis. We can do the analysis as many times as you need to, which means in some situations, some projects, it's very straightforward. It's not necessary to have an ongoing conversation about the project because you just want to identify a protein. But in some situations, we actually want you to come back to us and say, right, so I want to see exactly what are the unspecific binders of my IP. And we can do a slightly different search. You can actually mark these unspecific binders onto your results. So you can see, okay, these ones are not interesting. So there's several things we can do. So a good analysis, actually, you come back to us, right? Can I see these? So, and we want you to do that. So there's, in no situation, we don't offer support for that. So this ongoing communication. So there's obviously lots of things that these softwares do behind the pipelines, but the most important thing that I want you to have an idea is the levels of information that you have in PD. So I already spoke a little bit about the peptide spectrum matches. That's when the software identifies a peptide or matches a peptide to your spectra and attributes a score to it. And then the peptides, the information coming from the identified peptides can come from different charge states. It can come from different modifications. So you need to combine this information into the peptides. And then you can combine also into peptide groups, which can have different types of modifications as you want. And then these peptides will belong to something, will belong to a protein. That's when you combine the information coming from the peptides into a protein. And actually, there might be a subset of proteins that have exactly the same peptides, which form the protein group. In ideal world, in an ideal situation, we'll have many unique peptides. So these are peptides which have an amino acid sequence that's unique to that specific protein. And that's no doubt that you have that protein. In situations in which you actually, the proteins have exactly the same peptides, what the software does is it will look at the number of PSMs, it will look at the length of the protein, and it will define something as the master protein, which is the protein that has the highest score and the highest probability to be the one that is justified by that specific group of peptides. So now we're going to go to the live session. So again, this will work better if you stop me at any time and ask questions. Okay. So let's start with the gel band analysis. Right. So when we send you an email with the results, you usually have a link to download your results, correct? And in this link, you'll have different data. You'll have an Excel spreadsheet, which usually we provide for you to look at your data in the Excel. And this is usually filtered information. So we already applied the general filters, which I'm going to show you what they are. And you can just look at it in your PC or in your Mac. So absolutely up to you where you look at the Excel spreadsheet. You have also several TXT files. The TXT files are completely unfiltered. This can be useful if, for instance, you have your R pipelines. So you can just use these TXT files and process your data with your own pipelines. There's many blogs available that teach you how to do this. In the future, we'll have support for this, so we'll help you with this as well. And there's also a PD result file for the projects that were processed in Proteome Discoverer. And this will allow you to use PD in your own computers. And that's a caveat. PD at the moment is only supported in PCs. In the future, we're going to have a couple of PCs in the facility where you can just externally link to them or just log into them when you do you can book the computer basically, and you can see your projects in there. Let's imagine that I've sent you some results and you want to load your results. You just come to this folder, load PD result file. And let's go to the folder where I've saved the PD result files. That's where the folder you saved your PD result file when I send it to you across to download. And obviously it's already open because sometimes it might take a while until you can open the project. And there you go. So the first thing, this is usually what you see in the first go, right? A very big page with lots of information. In some situation, information that's not useful whatsoever for identification because this is a simple gel band analysis. So one way One way to kind of limit this information, you have this window here, that's the layout selector. And if you click on here, there's already several factory or default layouts that you can use. Some of them you can create yourself once you get a little bit more proficient in how to use the layouts. You can create yourself, save it, and then next time around, you just open a project, click on the layouts, and then you go. So I'm going to use and I think actually the band analysis is one that I already created a very long time ago. So once you click on this new layout, it will filter off the information that's not necessary for this kind of analysis. And now, the next thing that I usually recommend you to do is just apply a filter set, because you have everything that the mass spec has identified, which means, for instance, there's proteins in there that don't have a very good coverage, or they don't sequence coverage. That's one way of filtering, or they don't have a very good score. So you can filter data. You can just filter to the proteins that are most interesting or most reliable to look at. And the way to do this is just you click on this filter that you have here and you load. And usually when we send the data, we also send a filter file. If that's not the case, then you can come back to us, say, I want to filter my data, but I don't know exactly how to do that. We can provide you one. So let's go to the gel band. And then once you load the filters, you can see that these are the filters that are being applied. So I only want to see proteins that are master. And these are the proteins that I spoke that are the ones that have the highest score within their group, are the ones that are most probably the ones that will be your identified protein. I want just to see the proteins that have a good FDR confidence. So I don't want to see proteins that we are not really sure that they are there or not. And this is actually not necessary for OpenID, so I'll remove it because we're not looking at abundances yet. Another thing that you can filter. So if you're happy with the filters, you just have to hit apply, and you'll see here in the bottom that the numbers will change. Right, so another filter that you can actually see, so there's a lot of keratins and there's contaminants as well. So one thing we can filter is we can take off the contaminants we don't want, and that's quite common in gels. The reason why is not because of your hair, although everyone thinks, no, it's my hair that's falling on the gel. No, it's actually the dust that's on the air. There's always dust around us, and most of the dust is actually our skin cells. So, and they fall everywhere. And when you're cutting your bands to do your gel band analysis, they just usually come with dust. That's what it is. So that's why we see a lot of keratins, and it's very difficult to control for this. So the only recommendation that we have to limit keratin is try to use everything new or very well washed recipients when you're cutting your gel bands. Make sure that they don't have any dust, but it's very common to see keratin in gels because you have to handle the sample quite a lot. So you can filter off contaminant, but I know for sure that there's some keratins that are going to escape this rule. So what we can create is a description filter where we say the description does not contain keratin. And then we can hit apply. It's going to filter your data. There you go. So these are contaminant is false. I'm just looking at the contaminant at the moment. So you actually see, you can reverse any of the filters that you do in your data. You can reverse and hit apply again. So it updates. One other thing that this is again a back and forth conversation between us and the user. For instance, if you want to identify or you're more interested in identify proteins from a specific complex. Look, I just want to see if these proteins are in there. You can give me whatever else is in there, but I really want to see if my proteins are in there. You can just provide us with a sequence, and what we do is we'll create this as a marking database. And you can actually see here on this right column that the proteins that are marked in blue are actually the proteins that we were asked to look into. And if you just want to see what are these proteins, you can just go here, mark as in protein database, complex proteins, and it will filter for those. at a moment, I'm not interested in this, so I want to see something else. So let's just remove this, apply again. And in some situations, it might be interesting to see what's the abundance between the different proteins. Generally, for gel band analysis, it's not necessary, but you can do. We provide gel band analysis in two different softwares. Scaffold is usually the one that allows you to see very straightforward. Is it there? Is it not there? But if you actually want to have a little bit more information about the quantification of specific proteins, you might want to go back to us and say, do you mind running it in PD? Because I do need to see what are the most abundant proteins. And I usually recommend that we do this in PD instead of Scaffold. And that's just because of the different way that Scaffold looks at the data to establish a quantification value. So in PD, there's a column that's called abundances. And if you by any chance want to sort proteins by abundance, it's very easy. So you just have to expand the column that says abundances. If you click on one sample or on the other sample, you see that it turns bold. So if you select this sample and then out of this box, you collect one time, it will sort descending. So basically the most abundant protein on your gel band will be this. And then you can do the same for the next gel band. So you can just bold it by clicking on it and do exactly the same thing, but twice. So now the top hit on your list is the most abundant protein in your second band. Okay, any questions? Anything you've understood how to apply filters, how to look at your data? So it's always here. You can always see what filters you are applying. It's very easy to change them. And basically anything that's here on these columns can be applied a filter, and anything on these columns can be applied a filter. So it really depends on the question that you're asking your data. In this situation in gel bands, we can look at specific proteins. If you know, or if you think that you are targeting for specific proteins, you're purifying a complex, you want to know if they're there, we can use this information. So if you give us the sequences or these sessions, we can use this information to mark the proteins. Again, this is a back and forward conversation with us and you. Let's move on to the IP set. I'm going to close this one. And to open a project again, it's the same thing. You just have to click open a PD result file. You just navigate to the folder where you saved the data that we've sent through a link, and you open it. Okay. So usually the same steps. This analysis actually, this data set was done using DIA. So it was not DDA, it was DIA. But you can see that we still process it in PD. If we were to use DIAN, or if we were to use Spectra, you would just have to receive results in a completely different way. So the good thing about PD is that you can actually see it. Once you learn it, you can see all your types of projects in the same platform. Again, this will change in the future because hopefully we'll have a bioinformatician that will support you much more and much better. It will do a customized analysis to your sample. So you can just come to us with questions, and you will run the pipelines for you, not on PD, potentially on R or whatever pipelines this new person is going to implement. Anyway, IPs. IPs are slightly different, right? So you have your control, you have your bait, and usually what you want to see in IP is proteins that are not present in the control because those are game-specific binders. So that's a slightly different question. So on this pane, the first thing that we're going to do is the same thing. Let's change the layout for an easier way to look at. So we can do quantification IP. It will become slightly easier to look at. Here we go. We can apply the filter set. Let's load it. This one, standard filters. This is usually the filter that I sent to you, and we hit apply. Let's just see. No contaminants, does not contain keratin, great FDR, and it's a master protein. And we hit apply. And always in the end, it's very difficult for you to see, but here, in the end, you actually see what are the numbers being filtered for. So you can see the whole if the data set has 2,000 proteins, when you hit apply, the first number that you see on the left is the number after you apply these filters. Even if you have a good computer, sometimes it takes a little bit of time for the filters to be applied. So the good thing about PD is also that it offers a lot of visualization tools that are inbuilt into it. And the way to access these, we can now close the filter so that we have a little bit more space. So there's an icon that displays a PCA plot. If you click on here, a new window will open, and you actually see that there's many ways here that you can play your data. You can play with to display your data. This one particular is just the overall sample abundance, how it changes between samples. And in an IP set, exactly, in an IP set, usually we don't normalize. And the only reason why is the way that the software does the normalization is it looks up the overall abundance of everything, and it chooses the proteins that are not changing to see what are the correction factors that it needs to apply to each individual sample to normalize. Where in an IP, you're basically depleting the sample, and there's a possibility that the proteins that your IP all are changing. So normalization is not advisable when you don't have a set of proteins that you know are not changing in an IP set. Having said that, if you know that you have housekeeping proteins that will be there, and they're certain that they will not be changing, we can use these. So we can create a customized FASTA that includes a subset of proteins that we know it's not changing, and we can use these to normalize the data and get this. Sometimes this can be a bait protein. I generally don't advise this because it's just one protein. It's just a subset of peptides. It doesn't matter if you have 100 PSMs, it's literally you're normalizing your sample to one protein, and that can be dangerous. In most of the situations, the variability that comes from an IP, it comes from the whole workflow that you're doing. So basically, if you control really well the input that you use to do the IP and the amount of beads that you are using to do the IP, make sure that you're using exactly the same amount of beads, and this is quite important. Usually, you can control this variance quite well. And that was the case. This is absolutely no normalization applied. And you can see that the overall protein distribution abundance is very similar between all the samples. That's exactly what you want. Right. Another cool thing that you can see, you obviously can see the PCA plots, and you can see that they actually group quite nicely. So there's one here, and there's the other group there. You can also see the volcano plots. And then one thing that you can actually see, or you can actually do in the volcano plots, is you can select, if you click with the right button on the mouse, and I'm doing it on touchpad. So if you select all downregulated proteins, it will mark the proteins here under this checked column. This is actually the non-specific binders, because these are the proteins that are more abundant in your control. So if you want, by any chance, if I was to do a follow-up analysis on this, I could use this information. I can extract the FASTA database and use these to mark on a follow-up analysis. I can use these to mark them as non-specific binders because they are more abundant in the control. Now I want to see what are actually the proteins that are more abundant in my IP. So I'm going to uncheck all visible points. You will see that they will lose the blue highlight, and you can just check all uploaded proteins. Now, okay. So you can then expand here, and you can sort by the abundance ratio. You can see that there's a lot of proteins in here. I have 100, right? What does this mean? We can use now this new visualization tool, which is a histogram, in which you can plot the abundance of that specific protein that's selected. You can right-click on the mouse to show ungrouped abundance. And this is actually, you can see that this is a protein that has a couple of missing values, right? So one filter, you can go back to your filter sets, and you can actually say on abundances, We can say that I don't want to see proteins that are only present in one sample or two samples. I want to see proteins that are present across all the three samples in my bait. So you can say, they might have any abundance, I don't care. You can say any value in at least three samples. And you can hit apply. You can use filters to help you get rid of those proteins that might be interesting, might not, we are not sure. These proteins, the ones that have 100, you will not see them in the volcano plot because obviously these are present in absolute proteins, so you cannot calculate the p-value on that. So when you want to actually look at the really interesting ones, this is how you need to filter the data. You need to look at the proteins that have this 100 ratio, because these are the ones that are only present in your bait protein. And once you mark them, so you can, a way to, let's unmark these ones, uncheck all visible points. We don't want to look at this anymore. If you have questions, let me know. We can select for these proteins and you can check them all at the same time. A way to filter, to select all these data is just, you select them in a table and you can just go here, check selected in this table. And you can actually, you want to see where they fall in terms of the abundance. So there's this scatter plot here where you can actually plot the abundance. In this case, we want to do the grouped abundance of the bait protein. So your bait IP, you refresh, and you have now a plot that's displaying the abundances of all your important proteins because they are only present in the bait group. This is not very visual because obviously it doesn't follow a normal distribution protein abundance. So you can just go here and just change to log phase. And now you can see that this is the bait protein. This is the most abundant protein in here. And the way to look at this is actually you can double click on the dots specifically, and it will highlight on the table which is the protein that you've clicked on. And everything else is the ones that are only present on your bait group and on the control sample. Right. What else can I do in an IP? One thing that's quite useful, I might show you this already in here, but it's probably more useful in the, we can go here, in the total protein amount. So PD also does an enrichment analysis. So you can look at the Go terms that were identified in your proteins, the Go terms for your proteins, and you can see if there's any specific enrichment for any Go ontology. And a way to do this is, let's uncheck it. Do you want to have this check? Let's keep this check because this is actually quite important proteins. Let's check also all upregulated proteins. So now everything is checked in this board. And one way to pull out the enrichment analysis is this tab here. And it will let you know everything on here. Let's just, please. It will let you know if there's any specific enrichment. In this case, there's a very obvious enrichment. I'm gonna go, yes. Where did I click? Here, on this icon over there. Then a plot as well. I mean, in the beginning, there's so many icons you can click on. The best way is just over the icons to see what information you can look at. So it will tell you what it is. You can also, I can show you this actually. So a nice way to look at your data to see if there's any pathways being up or down regulated is use this enrichment analysis. Right, let's tick this off. And you can also, you see this pane over here, show associated tables. So this is, do you remember in the beginning when I told you the data is organized in different, so it starts from PSMs to peptides, peptide groups, proteins, and protein groups. So this is a way to actually look at this chain of information that's related to one another. So if you pull this, let's say, to the base, uncheck all visible points. Let me see if it's, so if you pull here associated tables, you have then 3 other tables that have associated data to these. So on this protein, I actually want to know what were the peptides that were identified. And in this situation, it's just one. Again, you can go back here. I'm not interested in proteins that only have one peptide. I'm not sure if the quantification is good or not. So on proteins, you can include a different filter. Let's explain. There's one that says unique peptides. And we want to see proteins have at least 2 unique peptides. So it's greater or equal to 2. Hit apply. One. This one has 2. More. Right, so these are the peptides that were identified within your proteins. Now, you might be interested in, right, I want to see how many peptide spectrum matches. You can actually see here, if you show associated tables and you go to PSNs, you can see that these peptides had these many matches. This is a different spectra. Each of these entries is coming from a different spectra. So it can be something that has been repeated, fragmented, because this is DIA analysis, so we are not excluding anything actually. Or it can be that it was different charge states. So what you might want to do, if you're interested, you can then go here and see the fragmentation spectra. So PSM is peptide spectrum match. So what it stands for is when the software does the match of your spectra to specific peptides. That's the first level of matching information. And what we will do is we'll attribute the score to how well your peptide has been matched to that specific score, that specific fragmentation spectrum. There we go. So this is how it looks. It will show you your fragment spectra, which is this one. And it will also show the theoretical fragment spectra. And this is because I'm using a tool that uses, which is called Chimeras, to do the DIA identification. So it does a theoretical fragmentation spectra that includes not only the masses of the fragments, but also the intensities. This is quite a complex tool. But you can also see what were the fragments that were assigned to that specific spectra that belong to the peptide that's supposed to be there. Now, this fragmentation spectra, you can see that there's a lot of things that are unassigned. That's normal because this is coming from a DIA analysis. And this is where the acquisition mode in which you actually fragment everything within a mass over charge ratio window, which means that you actually have a lot of peptides in there. Right. So let's go back to the IP set. I don't think I have anything else. Do you have any questions of? So the 100 ones are the ones that you don't actually see on the controls, and that's very straightforward. But you might actually see one protein that's present in the control, and it's present in your bait as well. But the difference in the abundance is so big that I would probably be very careful in saying that this is an unspecific binder. Probably is not. It also works as an unspecific binder, but it might be more specific to your bait protein than as an unspecific binder to your control, to your GFP or whatever you're using to do the IP. You can actually look at an example of this. Right, this one. Let's see how it looks. See, this is an example. It has been identified in both. It has abundance in both, but the difference in fold is so big, I would probably not say that this is an unspecific binder. It also binds your control, so probably it's binding something else that's in your antibody, your GFP or whatever. But look at the difference in abundance. I would still say that there's some specificity to this to your bait protein, so be careful with this. Don't assume that only the ones that are not present are specific. It might not be the case. Any other questions? So it's basically another way of, in those situations that you ask, where you just have n equals 1, we can still do a mimic statistical analysis. It won't be perfect at all because usually we want n equals 3, but we can try and do some analysis for you if you only have n equals 1. Right. Let's get rid of these guys. I'm gonna get rid of these. You've done a TMT experiment and you've received your results. Again, it's the same thing. Do it again. Load the PV result file, go to your folder, total proteome, ESTKO. This is actually a standard that Thermo uses for us to validate our instruments to see if the instrument or specific components of the instrument are working as they should be because we are doing a TMT analysis. But it's a good way also to look in terms of what we can see in the analysis pipelines. ESTKO is basically they have 3 different strains with the knockouts. They've knocked out 3 different proteins. And what you want to see is exactly how is the software looking at these proteins? Are they really being knocked out? So this is the type of sample that we have in here. Right. So again, the same thing. You might want to filter for quantification or change the layout for quantification so it gets less complex. You can always change back to different layout settings. So if you prefer to do the pre-analysis or you're filtering in a narrow way of looking at the data, you can do so and then revert back to a different layout afterwards. That's absolutely fine. So change the layout. You have your abundances in here. You have your abundances grouped. So basically this is the average abundance within your groups. And you have also the group CVs. That's quite important, especially if you're looking at a whole proteome analysis. You might want to get rid everything of the proteins that have above 30% coefficient of variation. They're changing quite a lot. So probably your statistical analysis won't tell you anything on that. So you can filter on that as well. Right. So filters. It's the same thing. We usually provide you with a filter. If not, come back to us. I don't have my filter, and we'll send it to us. See? And then, so let's look at the filters. Sure. Yes. Yes. So basically, when you hit your load, you can copy across the file that I've sent you onto this folder. This is the endogenous folder where PD saves the filters. And you can just, once you copied it across, you can select it. That's it. Yeah. I mean, it's not so much for reproducibility. It can take a while until you put in all the filters that you want. So you kind of want to start already from a baseline, right? You can build on that. So what we provide is usually the basic filters. And then you can build depending on the questions that you want to ask your data. For instance, in some situations, you might not want to filter out proteins that came only from one unique identification, from one unique peptide, for some specific reason, because actually your protein of interest is really low in abundance. You just want to see if it's still there. So if I was to apply any of these filters and then provide you with the data, with these filters applied, you might be missing out on information. So this is why I usually send the basic ones, but I want you to understand enough how you can apply these filters because you can play with the data, right? It is really important that you take ownership of the data yourself because the more questions you ask, the more questions you know how to ask to the software, the better you can grasp how good or how bad your data is. So there's a level of empowerment that I think is quite important. It's going to be really useful for us to have a bioinformatician because there's so many things that these softwares cannot do. And I can give you already an example. The normalization is something that we only have 2 choices. We can either do based on the whole background of the proteins that are not changing, or we can use a subset of proteins that we know it's not changing. There's so many more ways we can normalize the data, but we cannot do it on the software. So this is where actually having a bioinformatician that can do this for you with different tool sets will be very, very important. And we will once more empower you to have a better outcome from the data you already collected. But to some extent, A bioinformatician will probably not help you if you have a very straightforward question that you want to ask. You might be the one doing this for yourself, not relying on a bioinformatician to answer a very basic question. So it's kind of a way to empower you to look at your data. Again, there's different softwares, and there's Maximum is great and pursues this kind of this visualization tool that PD has inbuilt. Perseus is the tool that allows you to visualize data that comes from MaxQuant or even from PDTXT files. But again, this is just options that we can provide to you, and you can have to look into your data. Right. So filters. Here I'm actually using a filter. So because there's two different levels, once more, this is a quantitative project on the proteome level at the hist, these three strains that have been knocked down from different proteins. So there's two levels of information. Remember, identification and quantification. In this situation, you could care less about identification because you're looking at quantification. So you just want to say, and because what our mass spec does is it identifies and then it triggers quantification. And in some situations, the quantification is really low. The signal to noise is really low. So we kind of want to look at that. And we tell the software, anything that has a signal to noise below 5, 10, arbitrary number, just don't use it for quantification because I think it's not going probably to be very reliable. So we say that to the software, which means that you'll have identifications without quantifications. So with this filter, what you're saying is, I just want to look at proteins that have an abundance attached to it. Otherwise, I don't care if it has been identified. And that's it. We hit apply. And you now have around 1,700 proteins. Right, so let's pull out the plotting options. Now you have the volcano plots. And you can see this is a very poor or not volcano plot. Right, I only see two proteins here. One up or down. Let's look at it. This is one. And this is so this is actually because I've marked, you remember that I've told you that we have the function of including a certain subset of protein sequences as markers. I've already done this to make my life slightly easier. So I've marked the proteins that have been knocked down. So you can see here, this is one of the strains, and you can actually see, so let's look at the protein abundance across the other strains and see what's happening. I usually recommend for you to see the unknown group abundances, and the only thing you have to do is right click and select unknown group abundances so you can see how it varies across the different biological replicates. So this is one group, second group, the other group, and your wild type. So you can see that this protein has been knocked down. How good, how bad, we don't know that. There might be a reason technically, there might be a reason why we're actually seeing a value in there. And this is basically related to the mass spec itself. So when you're selecting to do the peptide identification and then the fragmentation of those to see the reported abundances, in some situations, we'll have co-isolation of different peptides, which means that you think you're isolating one specific peptide, but there's a couple of them also fall very near in terms of the mass over charge ratio. And when you select them to do the quantification, they also get fragmented. So you have the TMT values or the report ions coming also from these peptides that were co-isolated. And sometimes this is what happens. So this is one example. Let's see, because I told you that I've marked these proteins to make my life easier. We can now sort these and we can see here, there we go, the second knockout and the third knockout. So this is a way to look at your data. Right, we can get rid of this one. Let's pull a peptide that has, you see these peptides here, for instance, these peptides are shared between different proteins. So we just tell the software, do not use, it's an option, you can use them, but we usually say don't use shared proteins because if we have a peptide that's shared between the proteins and you are using this to add to the quantification value of one protein, how sure are you that this peptide does not belong to the other one instead? So we usually just use unique peptides to do the quantification. So let's associate tables, the PSM, let's see if it works. If you by any chance, I've never tried this before, I've never gotten this error because I usually do it from the endogenous file folder that I've searched the data. If by any chance get this error saying that you cannot view the spectrum file, that's probably because I've sent you the PD result file. It does not see where the spectra are. Let me know. And if you want to have a look, come on down because potentially you have to look at. I just wanted to show you the reporter ion. You can actually see the reporter ion coming from the spectrum for that specific PSM. Let's see. There we go. So this is the reporter ion abundance that was used for the quantification. This is coming from the spectra itself. According to what I've just done there, this is probably something you cannot see with the PD result file that I'm sending across. So if you ever want to look at this, let me know. So this is actually, do you remember when I said, right, we do the MS2 for the identification, we see the fragments of the peptides, establish a PSM coming to it. What the instrument then does is go through another round of fragmentation. It selects the 10 most abundant fragments and the ones that still have the reporter ions attached to it to do this third round of fragmentation. The only reason why we collect data this way is so we minimize the co-isolated peptides, so we don't have this interference coming from peptides that are not the ones coming from our protein. So when you do the third round of fragmentation to get the reporter ions, this is exactly what you see. You can actually see, so this is why you need a high resolution mass spectrometry. You can actually see that two of the reporter, there's pairs of two sitting very close in terms of mass over charge ratio. If you can see that over there, but it's really, really close. So when you're working with TMT, you do have to use high resolution in order to make sure that you're looking at the right reporter ion. Right. Anything else? Do you have any questions? Anything else? Don't worry if you don't. Whenever you have your result file, whenever you don't understand something, whenever you actually want to apply a filter and don't know how to get to that, let us know. Come on downstairs, we can help you. Okay? If you, I know that there's a lot of people that have problems because unfortunately PD is PC bound. We'll have a couple of PCs in the facility where you'll be able to log in, work with your project, and then log off. So this is on the way. And eventually very, well, towards the summer, hopefully if everything goes okay, we'll also have a bioinformatician with us that will help you to get more out of your data. Again, I don't want the bioinformatician to be doing this very basic stuff for you. I think that there's a level of empowerment in you being able to look at your data and extracting basic results. But if there's a complex analysis that needs to be done, the bioinformatician will help you do this. And in the future, one of the things that I also want to do is we'll turn this into a workshop in where actually you can do it at the same time as I'm doing so that we have exercises. But there will also be, hopefully, workshops, which include our pipelines, where you can actually completely disregard PD or anything, and all the analysis is done by yourself from the PSM level. So there's a lot of analysis steps until you get to the protein abundance and protein quantification, but we can get you there. Okay. This is always open. We are always open for questions. PD is a powerful tool, but it takes time to learn it. Okay. So that's it. Thank you.